So throughout this class, there are three key segments that we're going to focus on. And the first is Foundations of Neuroscience. Today, we're going to talk about the past, present, and future of neuroscience and the history of this field. We're going to talk about neurons and neural communication, neurotransmitter systems, and the structure of the nervous system. This is a really cool chapter because we're going to learn about how the brain develops. Then we'll move on to brain and behavior, which is personally my favorite chapters because we get to get into the really cool, exciting research in biological psychology. We'll talk about chemical control of the brain, motivation and eating, sex, emotion, sleep, language, consciousness and attention, and mental illness. And finally, we'll talk about the changing brain and neurogenesis, which is the creation of new neurons and brain cells, synaptic pruning, synaptic plasticity, and the neural mechanisms of learning and memory. So first, we'll jump into chapter 1, which is neuroscience, the past, present, and future. And I do want to note that although I talk about neuroscience a lot, biological psychology is essentially synonymous with neuroscience. It is the study of the brain and the central nervous system and peripheral nervous system. So today we're going to talk about early views of brain function and their importance, how ventricles were viewed throughout the ages, nerves as wires, what neuroscience and biological psychology looks like today, and animal use and research. It is super interesting to look at the evolution of the human brain. So here we're going to talk about some prehistoric views of brain function and importance, but first thought I would just show you brain size throughout the last several 100,000 years. So for the time of Homo erectus to the Neanderthal man, there's been a rapid increase in brain size. This actually occurred about 500,000 years ago when the human brain tripled in size. What's really interesting as well is that communication by symbols occurred about 320,000 years ago. So the modern human brain is the largest and the most complex of any living primate. I recently visited the Natural History Museum in Washington, D.C., and the Smithsonian has an incredible display of fossils of more than 6,000 different individuals that have been discovered throughout time. Some of these fossils are actually over 1 million or 2 million years old. And you can see how brain size has changed based on skull size. So as you can see, the skulls of the Homo erectus man to Homo sapiens has changed quite a bit. And this is due to the tripling of the brain. Interestingly, even in prehistoric times, there is evidence that they understood the importance of the brain. So in Neolithic sites, they have found skulls that are less than 10,000 years old, and there's evidence of head injury in early hominids. In this picture, you can see that there are holes in the skull, and this was due to a process known as trepanation. Trepanation is the procedure in which holes were drilled in the skull while a person was still alive. This was a prehistoric surgery that was most likely conducted due to maybe chronic headaches or migraines, or perhaps they thought they could release bad spirits from the brain, when in reality, this man could have been potentially struggling with mental illness. So we know that this man was alive during the surgery because there is evidence of regrowth of cellular tissue where the drilling of the skull occurred. This is a Peruvian tummy, and it was used during the trepanation procedure. So you can imagine how brutal this prehistoric surgery would have been because they used this tool to drill holes into the brain. We're now going to talk about ancient cultural views of the brain in Egypt, Greece, and Rome. And it's interesting. For example, in ancient Egypt, the Egyptians did not believe that the brain was very important. In fact, they believed that the heart was considered to be the seat of the soul and the repository of our memories. They thought so carelessly about the brain that during the embalming process, they would actually scoop the brain out through the nostrils and disregard them while preserving and embalming the heart and other organs in the body. As you can see here, these are the tools that the ancient Egyptians used to simply scoop out the brain out of the skull and discard it. This was an image taken by archaeologists and scientists when they were conducting imaging to look within a mummy, and they found that a tool was actually left behind during the embalming process. And now we'll move on to ancient Greece, where they had several different views about the brain. Ancient philosophers such as Hippocrates and Aristotle tried to create theories of the purpose of the brain. For example, Hippocrates, who is the father of Western medicine, believed that the brain was not only involved in sensation, but was the seat of intelligence. This was contradictory to other philosophers' viewpoints, such as Aristotle, who believed that the heart was the center of intellect, similar to the ancient Egyptians. Interestingly, Aristotle thought that the brain served as a cooling agent, sort of like a radiator for blood going to the heart. And now we'll move on to ancient Roman views of the brain. In ancient Rome, there was a famous physician. He was a Greek physician named Galen, and he was a physician to the gladiators. So you can imagine some of the terrible spinal cord injuries and brain injuries that he saw as a physician. He embraced the Hippocratic view of brain function and believed that the brain was the seat of intelligence. However, during this time, the church was not okay with scientists and physicians conducting dissections on cadavers or humans. So, most of his views of the brain were taken from his sheep brain dissections. Galen performed many sheep brain dissections, and when he conducted these, he found that there were two major parts of the brain. First, if you look at this top view, which is also known as the dorsal view of the brain, we can see that there are two key areas here. We have our cerebrum and the cerebellum. He suggested that the cerebrum must receive sensations, while the cerebellum must command muscles down to the body. And interestingly, even though he did not have much evidence for this theory, he was sort of correct. In fact, the cerebrum does help us process sensations and perception, and the cerebellum is our key center for movement and motor coordination. Galen also discovered the ventricles in the brain. He wanted to know how the brain received sensations and moved the limbs. So as he cut into the brain, he found that it was very hollow, and this large space within the brain is called the ventricles. These are openings in the brain that are filled with a substance known as cerebral spinal fluid, or CSF. So CSF is held in the ventricles, and as you can see, there are two lateral ventricles here, a third ventricle, and the fourth ventricle, which goes to the spinal cord. Interestingly, cerebral spinal fluid is super important because it cushions the brain, and 98% of our brain weight is taken away because it's floating and tethered in CSF. Could you imagine if we had to carry around large brains that were heavy and we had to support them? Our necks would be sore all the time. So this is the way it was designed to help prevent us from carrying that weight. However, Galen thought that the CSF played an even more important role, and he believed that brain injuries were not actually detrimental if they didn't penetrate the ventricles, which is more interesting considering that if you had a brain injury in the back of your head in the occipital lobe, where life-sustaining brain regions are, such as the pons and medulla that control heart rate and respiration, would cause a much more detrimental injury. So this theory was called the ventricular theory, in which Galen believed that the CSF played a key role in commanding communication between the brain and the periphery of the body. And so this theory that he had prevailed for 1500 years. And now we'll move on to the Renaissance era, in which the ventricular theory was still unchallenged. So the great anatomist Andreas Vesalius lived during the Renaissance, and he actually drew more detailed structures of the brain. During this time, as I said, the church was not okay with people conducting dissections on cadavers, so it was very difficult to obtain pictures, and drawings of internal structures. The ventricular theory of brain function still remained unchallenged at this time. So remember, this is Galen's theory that the CSF plays a key role in communication between the brain and the peripheral systems in the body. The ventricular theory was not truly challenged until the time of Leonardo da Vinci, who was one of the first people to study the nervous system and actually draw out anatomical structures such as the skull, brain, and cerebral ventricles. In the Middle Ages, there were many conflicting ideas, but Da Vinci defied the Pope at the time and examined and drew over 300 cadavers. During this time, there were black market bodies sold to curious scientists and physicians by grave diggers. So medical schools would actually steal the cadavers and look over them throughout the night, sometimes in the basement of churches even. And this could only be done at night because the church was not okay with them performing these dissections. So Da Vinci was one of the first people to actually map out a lot of brain anatomy. Interestingly, Leonardo studied the cranial nerves by creating a cast that showed the shape and extent of the ventricles. And he is one of the first people to actually draw out the ventricles of the brain. He identified the olfactory nerve as a cranial nerve, and through his dissections, he created a new theory due to the fact that nerves do not converge on the lateral or third ventricles, which you can read about here in this primary research article by Pevsner and colleagues that was published in 2019. Interestingly, this is one of the first theories that really stood out because it showed that maybe the ventricular system and the CSF fluid wasn't responsible for communication between the two nervous systems, but perhaps the nerves were playing a key role here. René Descartes was another philosopher who wanted to answer the question, where are the brain and mind connected? He actually thought that the mind was its own entity, that the spirit was a separate entity from the brain, and he was an advocate for the fluid mechanical theory of brain function, similarly to Galen, thinking that the CSF played an important role. Descartes believed that the mind was a spiritual entity that actually received our sensations and command movements by communicating with the brain via the pineal gland. Interestingly, if we look at the pineal gland, it sits in the very center of the brain. He believed the pineal gland was the seat of the soul. Interestingly, he created this hypothesis with no scientific evidence or backing. And the main function of the pineal gland is actually to receive information regarding the light-dark cycle through our brain's master clock. This is known as the suprachiasmatic nucleus. And so the pineal gland receives information from the suprachiasmatic nucleus and conveys this information to the pineal gland to produce and secrete the hormone melatonin, which helps us sleep at night. So Descartes' theory had no backing or evidence, and I think that he believed that just because the pineal gland was in the center of the brain, that it was the seat of the soul, when in fact it's really just important for helping us produce hormones like melatonin. In the 17th and 18th century, scientists started to break away from these traditional focuses on the ventricular system and started to look at brain tissue more closely. They observed that there were two different types of brain tissue, including gray matter and white matter. Gray matter is a collection of neuronal cell bodies in the central nervous system. So the cell bodies contain the cell's soma, which is also known as the cell body, and the nucleus that holds our genetic material. And throughout this class, we're going to learn more about that. The white matter is a collection of the central nervous system axons, which are large branches that come off of neurons and help them communicate with neighboring cells. It is important that axons can cross over between these two different hemispheres through a region called the corpus callosum, which we will also talk about more in this class. Another breakthrough in neuroanatomy was when scientists discovered that there were bumps and grooves in the brain. These bumps are now known as gyri, and the grooves are known as sulci or fissures. They can be identified on the entire surface of the brain. So why do we have these? The gyri and sulci play a really important role Because the human brain contains a very large surface area. We have a larger volume of brain tissue compared to other species. Thus, to make the brain more compact and to reduce the amount of surface area, the brain tissue is folded so that we can have a smaller brain size with more tissue that is more dense. You've probably heard of the term smooth brain. Sometimes, even on Instagram and TikTok, you'll see videos of pets doing silly things, and their owners will say they have a smooth brain. And that's because they do not have as much brain tissue and they don't have as complex cognitive processes that a human does because they don't actually have the gyri or sulci like the human brain. And we'll talk a little bit more in this class about disorders associated with the lack of gyri and sulci. So let's review how the nervous system was understood at the end of the 18th century. So even in prehistoric times, they knew that injury to the brain could cause death. And there is evidence of prehistoric brain surgery via trepanation. They figured out that the brain communicated with the body via nerves thanks to Da Vinci's drawings. They identified different parts of the brain. They hypothesized that these different areas perform different functions. However, they weren't able to actually study this in depth due to lack of technology, so there were only hypotheses about this during that time. They also hypothesized that the brain operates like a machine and follows the laws of nature. It wasn't until the discovery of electricity by Benjamin Franklin in the 1700s that scientists started to think about the potential electrical phenomena in the brain. Two scientists, including Luigi Galvani and Emile Dubois-Reymond, found that muscles can be caused to twitch when nerves are stimulated with electricity. And they figured out that the brain itself can generate electricity. So these discoveries helped the notion that nerves are like wires that help communication between the brain and the rest of the body rather than by the movement of fluid, such as the ventricular theory that was first proposed by Galen. The new concept was that nerves are like wires that conduct electrical signals to and from the brain. So essentially, nerves are wires that fire together. We're going to talk about this when we discuss action potentials later on in this class, but essentially, the nerves function to help communication between the brain and other nerves throughout the body. At this time, it was unclear which nerves were utilized by the body to serve different purposes. So there were two scientists, Charles Bell and Francois Magendie, who discovered two different types of fibers that existed in the spinal cord. Interestingly, both of them were competing against each other to try to discover the different fibers and their purpose first. So here we can see that the dorsal roots are red. The dorsal roots come from the back of the spinal cord, and they're in charge of processing sensations. Whereas the ventral roots, shown here in blue, are at the front of the spinal cord, and they help with the command of motor movement. So I do want you to know that the dorsal root enters towards the back of the spinal cord while the ventral root enters from the front. And so Bell wanted to understand how the spinal roots carried information in different directions from the brain to the periphery and from the periphery to the brain. So he cut only the ventral roots, not the dorsal roots, but he cut the ventral roots in a frog and found that it caused muscle paralysis. So this is how he discovered that the ventral roots play an important role in our motor commands. While Magendie was able to show separately in a different experiment that dorsal roots carry sensory information into the spinal cord. So ultimately, even though they were competing against each other, the Bell and Magendie law was created based on both of their separate discoveries. And this concluded that within each nerve is a mixture of many wires, and some bring information to the brain from the spinal cord, and then there are some that are needed to take information from the brain back down to the spinal cord. We do have 31 pairs of nerves in the spinal cord. I don't expect you to know what those nerves are in this class. If you wanted to learn that, you could take a neuroanatomy class. But remember that the incoming sensory fibers are red and the outgoing motor fibers are blue. And these are the ventral roots. Interestingly, other physicians and scientists have tried to discover how the different areas of the brain could be responsible for different mechanisms, such as personality. So, interestingly, there was an Austrian medical student named Franz Gall who tried to answer this question by creating a phrenological map. This is the study of phrenology. He believed that bumps on the surface of the skull were responsible for certain personality traits. Perhaps one area or one bump could help you study the personality trait of a person that was based on generosity or secretiveness or destructiveness. He thought that personality could be demonstrated through dimensions of the head on the skull. This science is called phrenology and it was debunked. It's false and it was never accepted by the scientific community. Interestingly, in neuroscience, most of our discoveries have happened by accident. So, localization of specific functions to different parts of the brain were usually solved due to brain injuries. For example, Paul Broca was working with a patient who had a stroke, and the person who had the stroke could understand language and could comprehend language, but could not actually physically speak. And so Broca worked with this patient, and after the man's death, he was conducting a post-mortem autopsy of his brain and found a lesion in the left frontal lobe. Here's the actual image shown here, and as you can see, there's a huge lesion, which means that there was brain tissue that was severely damaged in the left frontal lobe. So Broca concluded that this region of the human cerebrum was specifically responsible for the production of speech, but it was not a key region for speech comprehension, as the stroke patient could still understand what he was saying. Conversely, Carl Wernicke was working with a stroke patient who could produce speech but couldn't actually comprehend it or understand it. And his patient had a lesion here on the back left of the cerebrum. And so the area of the brain that's involved in speech comprehension is now referred to as Wernicke's area. And so in these patients that have Wernicke's aphasia, they can produce speech, but because they don't understand language and they can't comprehend it, the speech that they are producing is called word salad. It's simply a gibberish that doesn't actually have meaning or make sense. So they can produce speech, but they can't comprehend it. In neuroscience today, it's a growing field. We're going to talk about so many different facets of biological psychology and neuroscience in this class. It's very much an interdisciplinary approach where you can look at different facets like molecular, cellular, behavioral, or cognitive neuroscience. And what's really awesome is that some fields are coming together, like nutrition neuroscience, to look at the effects of nutrition and diet on the brain. I'm hoping that in this class, you'll find a passion for biological psychology and neuroscience. And even if you're not, that's okay. But there are several different specialties in this field. And I am an experimental psychologist and neuroscientist. And these are all of the different career options that you could look at if you're potentially interested in this field. If you guys ever have questions about grad school or your future career options, I'm so happy to talk about that with you. We're also going to be talking about the use of animals in neuroscience research and psychology research throughout this class. And I do want to note that research has taken a lot of steps to be extremely ethical. Now researchers have to adhere to the Institutional Animal Care and Use Committee. We try to use alternative methods, but we do need animal models for the study of disease. So we try to minimize any pain or distress, and animals are very well taken care of in research today to ensure that we are looking out for animal welfare, but also studying ways that we could protect humans by creating better therapies for disease. Some people ask, is neuroscience research worth the cost? Well, these pie charts here are showing the global burden of neurological disorders. This could be strokes, Alzheimer's disease, spinal cord injuries, etc. As you can see after a stroke in image A, this is looking at the proportion of disability adjusted life years after a stroke. So 42% of stroke patients are disabled following the stroke. This bottom pie chart is showing the amount of the percentage of deaths from different neurological or neurodegenerative disorders. Currently, Alzheimer's disease is the seventh leading cause of death in the United States. Additionally, many stroke patients sadly die from the repercussions of the stroke after living a disabled life. So, I think neuroscience research is worth the cost because we can create animal models to find more effective therapies and prevention strategies for this plethora of neurological and neurodegenerative diseases. And as many of these diseases do not have a cure or very effective treatment plans, neuroscientists can use animal models to find better treatments or therapies for our loved ones that are potentially struggling with these disorders.